possibility of removing disease from birth using gene editing technologies like CRISPR is one of the most profound advancements in modern science. Imagine a world where genetic disorders such as cystic fibrosis, sickle cell anemia, or Huntington's disease could be eradicated before a child is born. This concept, rooted in precise genetic interventions, holds transformative potential for human health, but also raises complex ethical, social, and scientific questions. In this video, we will explore the implications of using gene editing to eliminate hereditary diseases at birth, considering both the promise and the challenges of such a future. The science behind the vision. CRISPR and other gene editing tools allow scientists to modify DNA with unprecedented precision. By targeting and correcting faulty genes in embryos, it's possible to prevent genetic diseases before they manifest. The process involves identifying a mutation responsible for a disease, using a guide RNA to direct the Cas9 enzyme to the precise DNA location, and then either disabling the harmful gene or replacing it with a healthy sequence. For example, in sickle cell anemia, a single nucleotide mutation causes abnormal hemoglobin production. CRISPR can correct this mutation in embryonic cells, potentially ensuring the child is born free of the disease. Visualizing this, picture a fertilized egg containing a genome with a harmful mutation. Scientists introduce CRISPR components into the embryo, where the system acts like a molecular editor, snipping out the defective DNA segment and replacing it with a correct version. The repaired embryo develops into a healthy individual, free from the targeted genetic condition. This approach, known as germline editing, ensures that the correction is passed to future generations, potentially eliminating the disease from a family lineage. The promise of disease-free beginnings. The ability to remove disease from birth could revolutionize medicine. Genetic disorders, which affect millions worldwide, often require lifelong management, expensive treatments, or invasive interventions. Eliminating these conditions at the embryonic stage could prevent suffering, reduce healthcare costs, and improve quality of life. For instance, trials have already shown success in using CRISPR to treat sickle cell anemia in adults, and extending this to embryos could prevent the disease entirely. Beyond single gene disorders, future advancements might address complex conditions influenced by multiple genes, such as certain cancers or heart diseases. This technology could also reduce health disparities. Genetic diseases often disproportionately affect specific populations, and access to treatments can be limited by socioeconomic factors. By addressing diseases at birth, gene editing could offer a universal solution, assuming equitable access to the technology. The societal impact would be immense. Families would no longer face the emotional and financial burden of managing chronic genetic conditions, and healthcare systems could redirect resources to other pressing needs. Ethical and social challenges. Despite its potential, removing disease from birth raises significant ethical concerns. Germline editing alters the DNA of future generations, prompting questions about consent. An embryo cannot consent to genetic changes, and future offspring inherit these alterations without choice. Critics argue this could lead to a slippery slope toward designer babies, where parents edit traits like intelligence or appearance, deepening social inequalities. Visualizing this, imagine a future where access to gene editing creates a divide between those who can afford perfect children and those who cannot, exacerbating class disparities. Safety is another concern. While CRISPR is precise, off-target edits, unintended changes to other DNA regions, could introduce new genetic issues. Long-term effects of germline editing are unknown, as the technology is still in its infancy. Regulatory frameworks vary globally, complicating efforts to ensure responsible use. For instance, in 2018, a Chinese scientist's unauthorized editing of human embryos sparked international outcry, highlighting the need for strict oversight. Balancing hope and responsibility. The dream of removing disease from birth is tantalizing, but it demands careful consideration. Robust safety protocols, global ethical standards, and equitable access are essential to prevent misuse. Public dialogue is crucial to address fears of playing God or creating societal divides. Scientifically, ongoing research must refine CRISPR's accuracy and expand its applicability to complex diseases. In conclusion, the ability to remove disease from birth could transform human health, freeing future generations from the burden of genetic disorders. By editing DNA at the embryonic stage, we could prevent suffering and enhance well-being on a massive scale. However, this power comes with profound responsibilities. Balancing the promise of a disease-free future with ethical and safety considerations.
What do you think? Should we use gene editing to eliminate diseases before birth? Could this be the key to a healthier future or a step too far in playing with human life? Let us know your thoughts in the comments below.
Will shape whether this technology becomes a beacon of hope or a source of division. If you enjoyed this video, Make sure to subscribe to this channel for more content. Thank you.